I'm on the world-famous Wall Street here in New York City. Behind me is the New York City Stock Exchange on which Volition RX are quoted. Inside the building, they're about to start a special event, meeting all kinds of people from investors, potential investors, and also analysts and key opinion leaders. We've been following the team and catching up on their exciting news, both now and looking at milestones for the future.
So again, thank you all. I'll just quickly whip you through the agenda so you can see what's ahead of us and what to expect from the different speakers. Of course, the first step is myself, and then Jake, who needs no introduction, our chief scientist, will go through the basis of the nucleosome technology and nucleosomics. We very firmly believe that the epigenetic signals are very, very important, and probably as important as the genetic signals, where billions and billions have been spent. And we've been really pioneering that work, particularly in blood. So Jake will go through what our technology is, why it's so special, and how well we have the entire field covered. So as it becomes more and more important, we will dominate the field. Then Dr. Axel Imhoff there will also talk about one of our new projects. We've not only now just been labelling one structure on the nucleosome, but able to target all the structures through mass spectrometry and sequencing. And he can describe some of that process work that's going through now. An incredibly exciting part of the expansion we're doing now beyond the basics of the platform. But again, it's all in the intellectual property we have or our processing. Then Jason, Dr. Terrell, will go through introducing Nu-Q Veterinary, which again is a very, very exciting broadening of our pipeline. It turns out that there are nucleosomes in dogs as well as humans, so he'll go through the huge market potential and how we intend to get there. And then via Skype, I guess, Heather Wilson-Ribble from Texas A&M can go through the veterinary market and how we can be involved with them and really work together to launch what would be a very exciting set of products. And then I'll just discuss and sort of bring this all together and how this works with our platform timings and the path forward. And then we'll have a brief discussion if there's time, depending on how much we take up your time in these slides. We'll have enough time for a few questions chaired by Jonas. And then we'll have closing remarks from our chairman. So that's what today's looking like. And then those who want to, we can go down and watch the closing bell, and then come up here. We'll have an hour of a mixing session. If you want to ask any particular questions to any of the team, or any of the new projects, or our long-standing colorectal programs, that would be a very good time to do it. So as we always talked about, we're a very dedicated team who are completely committed to revolutionising cancer diagnostics throughout the world with routine blood tests. I'll just start with this slide. I think what this really shows our huge commitment in the last couple of years to developing the platform and really delivering on the promise that we've had, and then the fantastic technology. For that, we've really had to adapt the platform to be very robust and all the things which our scientific team can go through. That facility in Belgium is 20,000 square feet. It cost us, including the fit out, the back half, his laboratory, was about 1.8 million euros. An amazingly good deal, a vast majority provided by loans and support from the Walloon region. Our Chief Executive of Belgium is at the back. You can ask him questions about it. We have over 30 people working there now. Some of them you can see here. It's always important to remember, if you're a real company doing real technology, you need a large team of people doing on the coalface, actually doing the work. So as you can tell, most of the team in Belgium is female. Very good lab technicians, and the space we have is really cutting edge. When combined with our large robotic machines and our technology, it's really quite something to see. So we're very happy with anyone who wants to come and visit us in Belgium. It's a fantastic facility. It's about an hour from the capital. It's quite remarkable. It's really allowed us to go to the next level of developing a really robust platform. This is a Capital Markets Day, and our Chief Financial Officer is also here, so I'll just go through the brief financials. We had $13.4 million in the bank at the end of last year. We had, through strong support of our shareholders, 6.7 million of warrants exercised, left us with about 20 million as of the start of the year, minus what we've been spending. So, and we've kept the run rate amazingly stable, around 1.2, 1.3 million a month, which is a whole different level of what most companies do when they're running large clinical trials. We can do that because of our setup in Belgium, because of the huge support we have from our clinical partners, where we pay a tiny fraction of the cost for all the trials we run. And we keep things very lean, because ultimately we're all large shareholders and we want to see a good return. So we keep it very tight. So this, just to reiterate a couple of things before we turn to the actual speeches and presentations. One thing to keep in mind, this platform has been developed. Our core project is the colorectal space. We have very large trials still being collected in the area. It's taken longer than we thought we realised there's a huge amount of work to be done. That's why we've been serious in the last few years about the new facility and rapidly expanding the team. That work now is coming through. We've had the first assays that have come through and they've been very, very good, extremely reliable, very good accuracy, and we're in the process now of finishing those off, and the first results we had were excellent. So beyond the core colorectal work, we're really thickening with momentum now in lung, in the veterinary scale and with Nu.Q capture, and in a range of other areas which I'll go through when we're finished in the process. So it's a lot going on for a small company, and the reason we can do all these things on a very small budget and a reasonably small team is it's a platform, an absolute true platform, meaning 90% of the work is between all the different trials, all the different cancers, the dogs, capture is common work we have to do for all of it. So it's something we have done on a very tight budget and we're continuing to do on a very tight budget. We're not looking to massively expand at all. And just as a quick reminder, if you do find cancer early and have it removed, your outcomes are very, very good. We believe absolutely the only way of getting cancer really, truly beaten is to have 80 or 90% of people with a good screening test that can find cancer early, with a tiny blood as a routine test. And as far as we're aware, we're the only company that really has something that can be truly routine, truly done at a low cost. There's a lot of expensive, complicated tests out there in development. None of them will be truly routine in the way ours expect to be when they're products. So this is something very different. We're swimming in our own lane, and I think we can add a huge amount of value to the company, but also to humanity through better tests. So that's what we're developing. And I'll turn over to Dr. McAuliffe, Jake, who can start the process with talking through the technology itself, the base technology. Thank you.
Thanks, Cameron. So, as Cameron was saying, in order to make tests really routine and increase compliance so that everybody that should have a test in a screening age, for example, does actually get a test, the test really needs to be accessible to people, low cost, and not require them necessarily to go to hospital for a day just for that test. And if you look at most diseases, in order to diagnose them and monitor them, it is blood tests that are used. So, in diabetes and cardiovascular disease, kidney function, anything, most areas of medicine that you can think of, the first thing that you do is a blood test. And the reason is because it's easy, it's low cost, and doesn't involve you going to hospital for a day. On the other hand, if you look at what happens in cancer, the tests are, with the possible exception of PSA being used off-label, as it were, the tests are not blood tests. So if you want to have a look for a lung cancer, it's an X-ray or a low-dose CT scan. There's mammography scans, various other scans, or you can have an endoscopy process where somebody puts a long tube inside you with a camera on the end, or you can have cervical smears. So all of these things are very invasive and not low cost, and have compliance issues because they're unpleasant in some way. So our mission is to change that by making tests that can be run routine. So they would be, for example, like a PSA test, like a cholesterol test, where you can simply take a sample of blood and run it as a test for cancer. And the important aspects of the test for that are really that it requires a small amount of blood, and that it's low cost and can be done in large numbers, which Nu.Q can be, and that's what we're doing. So what's the basis of it's a big claim to make. So what's the basis on which we can do that? The basis is that cancer is really a disease of the nucleus. It's a disease of the DNA and the chromosomes. It's when the chromosomes go wrong and cells start to divide repeatedly and out of control that you get a cancer. This happens because of misfunction in the nucleus, in the DNA and in the chromosomes. And if you're going to look for a test, really that's where you need to look. So PSA, if you like, is the exception that proves the rule. It's a protein that's specific to prostate, which shouldn't be in the blood. And if it is in the blood, there's something wrong with your prostate. But in general, you're not going to diagnose cancer like that. You need to find that malfunction in the nucleus. And that's essentially what we do. Because when cancer cells die, these, the chromosomes are digested into the individual little balls in this picture. We've got a model of one here that Professor Axel made for us. And those individual nucleosomes originating from the cancer are in the circulation in the blood. And for quite a few years now, people have been looking at the DNA that goes around them, that wraps around the nucleosome, to see if they can find mutations in the DNA that relate to particular cancers and sequence mutations related to cancer. The problem with doing that is that there are about 20 million nucleosomes in the human genome, and that sequence will only occur once. So it really is, it's a 1 in 20 million shot. So you're looking for something that's 1 in 20 million in something that's already pretty small. So it doesn't work, extremely well. It works at late stage, it doesn't work early stage. And the difference with what we're doing is that we can look at all of the nucleosomes, because the epigenetic changes that occur in the nucleosome occur right across all of the chromosomes. They don't occur to one particular point. And the reason that happens is because they're enzyme based, and the enzyme doesn't look for a tumor associated gene, it just does the whole thing. And the course of evolution of the cancer does the rest. So our aim, and the project that we're doing, is to look for those cancerous, is to look for the nucleosomes themselves, which are elevated in cancer anyway, and also for the particular associated cancer changes. And here's a cartoon of a nucleosome, really just to show some of the potential changes that can occur. So each of these points that's marked here is a potential place where you can get a signal, an epigenetic signal. It's a chemical signal. And in many cases, there are multiple changes, multiple different signals that you can get. And the 4 proteins listed there, H2A, H2B, H3 and H4, they also have multiple isoforms. So for many of, there are 300 or 400 at least human different potential proteins that you can have there, and all of those are epigenetic signals as well. So there's a lot of things that we can measure and that we are measuring. And what we've done recently, I think probably you will have heard, and I've already been asked a couple of times today, we've recently revisited how we do this, and actually the platform is now really superb, and we can detect individual signals. We can prove that it's quantitative. We can prove that we detect only that signal and not, if that signal's absent, we can prove that we don't detect anything. And if there's a similar signal right next door, we can prove that we don't detect that. So the platform is absolutely superb now, completely reproducible and ready for product. So with that new platform, I'm very happy, by the way, to talk about the technical details of that. What's different about the platform now? Why is it so good? Probably not, maybe not, this is not the right place, but if anyone wants to ask me during drinks afterwards, I'm happy to go into that in detail. So we've now put some of these assays through some clinical studies, small numbers at first, but we will be moving it into the big numbers. And here's just some examples of the sorts of results that we've got. So in a lung cancer cohort, it's small numbers, it's 76 people, but with just one of our assays, we were able to get an AUC of 85%. And that was cancer versus healthy, but it also discriminated lung cancer versus other lung diseases. And then we looked in a second lung cancer cohort, that was 152 people, and did exactly the same thing and got a very similar results, about 80% AUC. So the platform itself is analytically reproducible in a scientific way, and the clinical results are excellent and reproducible. So we will be putting that assay together with others to increase the AUC further. In a separate experiment, we looked at colorectal cancer. That was 123 people, and with the same assay, it detected it had an AUC of 72%. But when we made a 2 panel, added one more to it, that went up to 84. And I've got a picture of what that looked like. So the green subjects on the left are people that were sent for colonoscopy, but actually they had a clean bowel, there was nothing in there. And then the 4 red groups are stage 1, 2, 3 and 4 cancer, and you can see very easily that we detect all of the 4s, and we certainly detect most of the 2s and 3s, and we even detect a very large proportion of the stage 1s. And actually detecting stage 1 cancer is something that there isn't any other test out there that does that. The Septin9 test, to the best of my knowledge, detects relatively few stage 1 cancers, and things like CEA don't detect stage 1 at all. And on the right hand side there's a ROC curve which shows, I don't know if you're familiar with ROC curves, but it shows how the sensitivity of the assay varies if you change the cutoff at which you do it. So these are early results. We will be taking those forward into bigger sets. In the lung cancer example in particular, we're now collecting 1,200 sample set of people that have had low-dose CT scanning. These are high-risk people that were sent for the best current lung test. The problem, well, the good thing about low-dose CT scanning, and really X-rays as well, is that it does find most cancers, because if you've got a lump in your chest and you X-ray your chest, you will find that lump. The bad thing is that it has a 40% false positive rate, because if you take, essentially you're taking a picture, and if there's a lump or a nodule in someone's chest and you take a picture of it, you can see it and you know where it is, but you have no idea whether it's malignant or not. And so 40% of the positives are false positives. There is something there, but it's not a cancer. And the study that we're collecting at the moment will be to see how well we can discern whether the people that have a nodule in their chest actually do have cancer. That's collecting now. We will get the first results of that early next year. And with that, one of the first part of any ELISA is to capture nucleosomes on the solid phase. But actually, once you've captured them, there are other things you can do than just ELISA. And one of the members of our scientific board, Professor Imhoff, is working with us on this new way of analysing nucleosomes that we capture. So I'll pass over to you, Axel.
Thank you, Jake. Just kind of stealing this. So, good afternoon, everybody. My name, as Jake said, is Axel Imhoff. I'm a professor at the University of Munich, and I'm the last addition to the scientific advisory board of Volition. And what I'm going to do for the next 10 minutes or so is to tell you why I'm so excited about the Nu-Q capture assay, and why I think that could really serve as an enabling technology for Volition to further improve the Nu-Q technology. As Jake mentioned, in ELISA assays, as the Nu-Q is based on, as the Nu-Q test is based on, what you do is actually you capture nucleosomes at the bottom of a plate out of plasma or serum, and then you detect one particular feature with another antibody that is actually labeled. And if you get label bound, then you get a signal. And if that feature that you're looking at is something that's specific for a tumor, then the signal you get is proportional to the amount of tumor cells that have lyse in the body. Right? However, this is one feature that you can look at. And of course, wouldn't it be nice to look at more features and to have more features, to have panels, so that would allow you to discriminate between different tumors and different tumor stages. And this is where the Nu-Q capture idea is actually coming from. So what we're doing is we're actually taking a similar strategy where we fish out the nucleosomes, but then rather than taking one antibody and looking at one feature, we actually try to look at multiple features, ideally looking at all the features that are possible in the nucleus that we fish out. And of course, I brought you here a 10 million increased model of a nucleosome. So this is how a nucleosome looks like. And what you immediately can see, there has two parts. It has DNA, that's the red bit, and it has proteins in the middle. So you can look at both features. Of course, the DNA, you can look at the DNA sequence and determine the sequence that you fish out. And the, in my opinion, more relative and more relevant and more epigenetic part is to actually look at the white bit, which are the proteins that make up the nucleosomes. And if you have some time later on during fish, you can actually look at this, and what you'll see immediately is that there are clearly more than one feature. And in fact, by using mass spectrometry, and this is what we've been doing for the last 20 years or so, you can detect up to 200 different features on the nucleosome in a or on the histone in a highly parallelized manner. So rather than in ELISA, where you actually look at one, you can look at those 200 features from one sample. The disadvantage is that you need more sample, and this is why we actually established this method where we moved away from the ELISA plate and used beads to fish out the nucleosomes. By that way, we can take larger amounts of plasma or serum and purify the nucleosomes from. And so in preliminary experiments, what we just simply did, we took recombinant nucleosomes and just checked whether we can pull down those nucleosomes from a mixture and whether we are able to deplete this. And in fact, we can do this. Sorry. Oops, no, it was too fast. Which you see here on the green bars, we add synthetic nucleosomes and we look at what is actually remaining in the supernatant, and you see that we can add quite a bit of nucleosomes and fish them almost all out and deplete it. That does not only work with synthetic nucleosomes, it also works with plasma, where we can fish out most of the nucleosomes out of plasma. And that, of course, now allows us to do nucleosomics, or to actually use those nucleosomes to detect features and learn something about the nucleosomes that we fish out of the plasma. And we can do this by DNA sequencing, looking at the circulating tumor DNA, and we can do this, and this is where my expertise comes in, using the histones and using mass spectrometry. And so the preliminary experiments told us that we were able to immunoprecipitate the circulating nucleosomes from plasma and serum, and we're now able to extract the DNA, and the goal is ultimately for the sequencing to look at the circulating tumor DNA. Now, what we have done is we actually separated the DNA from the proteins and looked at the proteins and did mass spectrometry in this. Indeed, we could already find more than just one epigenetic feature in those circulating nucleosomes, multiple modifications, multiple variants that are all potentially indicative for different types of tumors. So just to summarize on the Nu-Q technology, we're actually able, and that's what we can show in those preliminary experiments, to pull out nucleosomes from a highly complex mixture such as plasma or serum up to a depletion of 90%. The next step is, of course, now to go into tumor samples to see whether for particular tumors we see particular features, and then ultimately to allow Volition to actually develop antibodies that can then be used in the ELISA platform, ideally in a pattern, so that you can actually really distinguish different types of tumors. So I'll just skip this here. The mass spectrometry is clearly an enabling technology in a shotgun way, where we can go one step ahead and then try to do some sort of biomarker discovery where on the nucleosomes that serve as biomarkers clearly for tumor, we can detect different features, and those features on the nucleosome will then allow us, or allow Volition to develop specific antibodies that one can use in multiple assays and then further and much more precisely do the diagnostic tools. I should say that this is clearly an R&D project, right? So we're still at an early development. We now have shown a proof of principle where we can isolate nucleosomes and purify them and do the mass spectrometry on them. But there's much more things to do, like looking at real tumor samples. I think those results are very exciting, and I'm super excited to work with Volition on this particular topic. And one thing I would like to mention while I have this here, this nucleosome, and this is what brought me into the nucleosome research, is highly conserved from yeast to man. So essentially the nucleosome looks like this, not only from humans, but also from animals, and I guess this is why Volition decided to start the Nu-Q-Vet program, and with this I'm heading over to Jason Terrell. Thank you. I'll leave this here.
Well, thank you, Axel. That was great. For those who don't know me, better? Yeah. For those who don't know me, my name is Jason Terrell. I'm the Chief Medical Officer for Volition and the Chief Executive Officer for Volition America. And I'm going to be introducing today Nu-Q-Vet and talking about the very exciting work that we're doing in animals, a little bit about the business strategy going forward, and introducing some of the key individuals who are going to be helping us to make that happen. So we decided several months ago, I think we've demonstrated that our platform is very effective at diagnosing cancer in humans. So several months ago we decided to take a look and see if it was equally effective diagnosing cancer in animals, and specifically dogs, because that's the largest need and the largest market. So there was two main questions that we had to answer first of all. First is, are these healthy nucleosome structures conserved across species? And second was, are the cancer-specific modifications that are present on the nucleosomes also conserved across species? To do that, we partnered with Texas A&M University and did a collaborative study there. We chose Texas A&M because they're one of the world's leading institutions in veterinary medicine, and they've been fabulous to work with. They're extremely proficient in conducting their clinical trials and very experienced commercializing products. And also they're as excited about the technology as our entire team is. So the study we did, it basically answered those two questions for us, and the answer was yes. The normal nucleosomes are conserved across species, and the specific modifications that tell if this cancer is present are also conserved across species. So that when a dog gets cancer, it displays the same profile that humans do, which is very exciting. So that's going to be true for dogs, but also for cats and horses, and basically for all animals. So I think now the best thing to do is for me to introduce Dr. Heather Wilson-Robles. She is a professor at Texas A&M University at their College of Veterinary Medicine. She's a very distinguished veterinary oncologist. She is the assistant head of their research department, and she's the chair of comparative oncology. So she will be joining us online. We'll see.
Hello.
Good.
I have.
Yeah. Can you advance the slides, Heather?
Yes, sir. I should have control.
Okay, excellent. Yeah, take it away.
All right, great. Thank you so much for having me. It's great to visit with you guys today. I'd like to sort of talk to you a little bit about what we've been doing at Texas A&M. Here working with the Volition guys and our canine animals. So give me just a second. Get these slides moving. Looks like you guys are stuck looking at me for a minute. I'm going to try to move this on. I don't know what's going on.
If you'd like, Heather, I can advance for you.
Sure. It's not, there's a little bit of a delay, but it doesn't seem to want to advance. Oh, there we go.
Yeah, you got it.
Okay, great. Thank you. So one of the things I think that's really interesting about veterinary medicine and human medicine is that dogs and humans are actually quite similar when it comes to oncology. I think one of the things that people don't really realize as much if you're not in the field is that we actually see more cancer diagnoses in dogs every year than we even do in man. And I think the reason for that is that 25% of all dogs over the age of 2 are going to die of cancer, 50% of all dogs over the age of 10 are going to be diagnosed with cancer in their lifetime. A lot of that is because these dogs live in our environment, they share our food, they sleep in the bed with us, they do all those things, so their exposure is similar to ours. But in addition to that, we breed these dogs in a certain way to have a certain look or a certain behavior or a certain personality. By doing that, we also breed in other weaknesses that may predispose them to cancer. And so when dogs get cancer, they get cancer for the same reasons that we do, and they get the same cancers that we do. Brain cancer is about 6 times more common in the dog than it is in people. We see the mucosal or oral type melanomas, which are extremely aggressive, many-fold higher incidence of that in dogs than people. Hematopoietic malignancies like leukemias or lymphomas are actually really common and represent about 25% of the cancers that we see in the veterinary world. Cutaneous melanoma, we definitely see a fair amount of that, especially down here in Texas. Mammary carcinoma, in any unspayed female dog, about 1/4 of them will develop mammary or breast carcinoma. Bladder cancer is another common one that we see that is very similar in dogs to humans. Prostate, and then bone cancer, which commonly affects children, is about 10 times more common in the dog than humans. And so I think because of that, the market here to develop a test that could help us detect cancer earlier would be hugely beneficial. When we look at what the market is in the United States and around the world, you know, we've got, even the most recent numbers from 2018 suggest that there's even closer to 90 million pet dogs in the US. What we're seeing is a huge increase in the number of pet dogs as millennials sort of choose dogs over children and sometimes even dogs over marriage. And people are spending a lot more money on these animals, their fur babies, so to speak. And so we do see a really large market, the U.S. being the largest of the developed countries with a large number of pet dogs. And as was mentioned sort of earlier on the veterinary side, we have basically no reliable blood test that will tell us if an animal has cancer or not. We don't even have PSA or CEA or any of those types of tests to access. And so because of that, I think we have this huge unmet need, but also what tends to happen is dogs come in really late in the disease process and then have to go through a lot of procedures, like they mentioned before, CT scans and scopings and biopsies. And in addition to that, most of our patients have to be anesthetized completely, so under general anesthesia for those procedures, which increases the risk as well as the cost for clients. And so having an easy blood test that could be used when you suspect cancer, and then hopefully even as a screening test early on for certain breeds, which have a known elevated risk of cancer. Take common breeds like Golden Retrievers, Boxers, Rottweilers, Bernese Mountain Dogs. These breeds have an 80 to 90% chance of developing cancer in their lifetime. If we were able to screen them in an early disease setting like stage 1 cancer, our abilities to treat those and improve upon our survival times would be dramatically increased. When it comes to nucleosomes, you know, you guys have already heard about how we're able to pretty reliably detect these nucleosomes in the blood of humans with cancer and trauma and inflammation, and how these little bits of DNA, which kind of these clusters of histones with DNA wrapped around them like thread on a spool, how these are released into the blood at different levels with different diseases. And we're usually pretty good about being able to differentiate between those diseases based on what type of nucleosomes or the number of nucleosomes are present in the blood. When it comes to dogs, the same is true. And so what we see in dogs is that both with trauma, infection, as well as cancer, we see these elevated levels of nucleosomes in their blood, which kind of mirrors what we find on the human side. And also, as was mentioned earlier, the histones really are conserved from species to species, and so very similar, so the platform doesn't really have to be changed or altered very much from human to canine. Here's just a list of some really common diseases that we see in dogs, and a lot of these are going to be diseases that you recognize. Under the sarcomas, which is one of the most common tumor groups we see in dogs, hemangiosarcoma, which is akin to angiosarcoma in people, bone tumors, those are common. Non-Hodgkin's type lymphoma, again, represents about 25% of the cancer that we see. We also see a lot of sinonasal cancers, and then things like thyroid and bladder cancer, mammary, which is the same as breast, are really common as well. On the more infectious or inflammatory side, dogs, a lot of them are athletes. We see a lot of these cranial-cruciate ligament ruptures in them, which can induce kind of trauma or inflammation. We see a lot of inflammatory bowel disease. We have an entire lab at Texas A&M based on GI diseases in veterinary dogs. And so there's a lot of that. We also see a fair amount of things like skin infections, ear infections, as well as pneumonia, which often are bacterial or yeast infections. So what we've sort of done thus far is looked at a total of about 112 dogs from 3 different areas, Oxford, Liège, and then Texas A&M. And what we sort of found was that regardless of geographic location in these 112 dogs, whether they were control or cancer or inflammation, that we found nucleosomes, and we found elevated levels of nucleosomes in animals with disease. And so we are working to kind of further expand that cohort. So we've sort of been able to show already that we can find nucleosomes reliably in dogs in serum and plasma using the platform that exists. We're in the process of requiring this study set of 200 samples. So in addition to the 112 that we've already looked at, we've collected about 100 more samples, which we should be shipping to Belgium hopefully next week. And we're going to use those to sort of lock down that test panel that we want to use moving forward. And that will be used to generate our kind of research use only test or platform. That platform will then sort of be used to validate an additional 100 cases of affected dogs, as well as 100 controls. And we certainly have collaborations with a variety of universities in the US, which we get along with very well, who can help us collect those samples in a pretty quick manner. And then our hope is to sort of use the structure of the GI lab, which is a world-renowned lab at the university, or at Texas A&M University, to kind of help us get those samples in, process them. That infrastructure exists. They receive thousands of samples every week, and so that will help us sort of get that under control. And once that's sort of set up, our plan is to perform the USDA regulatory approval study so that we can actually get to a commercial launch. And so I think kind of the closing summary for that is that we've had really encouraging preliminary results. We have a massive market in which 25% of dogs will not only develop but even die of cancer in their lifetime, and half of all dogs over 10 are going to get cancer. In the US alone, there's at least 4 million cancer diagnoses per annum, probably a lot more than that. These are the ones that get reported or end up in specialists' office. And I think what's really nice about this platform is the owner's not committing to treatment, they're not committing to seeing a specialist. Before you do this test, this is a screening test that can be done in any veterinary clinic. The test looks like it should be very affordable within the clientele market. And again, we are working really hard with Volition to put this together. So we hope to register this project or product with the USDA, and that should allow for pretty rapid movement into the actual market, hopefully faster than what we see on the human side even, because the veterinary market's a little bit easier. And so with that, I will turn it back over to you, Jason.
Thank you, Heather. That was great. Let's see if I can take control back. Not yet. So I think you can see why we are so excited about this opportunity in veterinary medicine. It's a huge market meeting a very serious unmet need for oncologists like Heather, for primary care vets, all over the country, and for the animals themselves and for their owners. And I don't know if everybody read our press release today yet, but we signed a memorandum of understanding with Texas A&M, which basically says that they're interested in taking an equity position in a Volition subsidiary and working together with us to commercialize this product, in a new company. And what's so nice about that is, Volition, we focus on human medicine, human cancers. Texas A&M focuses on animal cancers. So we want to let them lead the charge of the research, the development, the commercialization, because that is what they do, and they do it time after time very efficiently, very effectively. And now you see a picture here of Nathan Dewsbury. Nathan is in the back. He's been consulting for us for many months, helping us define the business strategy, define the path forward. Nathan is a Texas A&M graduate. He has a very impressive resume, which includes successful research development and commercialization of in vitro diagnostic tests in all animals, including dogs. And he's also built from startup, himself and one partner, a very successful international pet care company. So with that, he knows all the bench work that needs to be done. He knows the regulatory pathways. He knows the commercial markets. He knows the industry. And we're very excited to have Nate as a consultant, and we're excited to have him working with us in this new company we're going to form in the future.
Have me again for one more time. So just a quick summary. I know there's a lot to process here, and it's even for us doing it every day, we think there's a lot of things going on. But I think what's extremely important to remember is all this is on the same platform, a very small budget, and it's all very much linked through what we do on the platform. So our focus, everyone asks what's our focus with everything going on? Our focus has been to make an absolutely world-class platform that can be used in a very wide range of areas very quickly around the world. And that's what we've spent a huge amount of time on. I know a lot of you see myself, perhaps Scott, a lot around the United States. You see our enthusiasm, you see, we think we're really doing great things, but what's really important is to remember behind any project there's a huge number of people. Any successful visionary project that is truly going to change what we do in a lot of ways and change the world, you need a very, very active team to do very different things in a whole lot of different ways. So I think you've seen some of the excitement from our team. There's quite a few other team members here. And I think it's also good for everyone to also see the people we work with on the advisory board and the processes. Very serious people who do very serious work, who are working very, very closely with us to make this a reality. And we can work very, very hard, but without the help of a lot of other people, including our partners. I'll just show you one slide now. This is just the trials we have collected or collecting in the different cancers, never mind a lot of the work we've been talking about today. So this is even more remarkable with a small team and a small budget and the focus we have on getting the platform. But all of these are reasonably easy to run once you have the platform developed. The very large trials being collected around the world and a lot of proof of concept studies, which, although they are small numbers, are meaningful. The statistical valid, we only use things which are 95% significant. So the smaller trials do tell us a lot about what's going to happen in the bigger trials. There's no point doing trials if they don't tell you, if they're just very small numbers. But when you're talking 30, 40, 50 cancers and up to 100 or 200 controls, it does tell you a lot of what's going on. So the vision has always been to get the platform developed, which has gone extremely well, continue to develop it. I think you've seen it's going to be, in our opinion, a whole new area of science and development. So it's not something we can do ourselves in a small number of years, but we've developed along the lines to get these things done. And by doing all of this, going through the small stages and the smaller trials this year, starting the larger trials and finishing them off next year, and expanding to a wide range of areas like the Nu.Q Vet, like Nu.Q Capture, and all the fantastic work being done in Asia should provide a huge opportunity for the company and therefore for shareholders. And just an order of magnitude where our market cap is now, successful companies in this space, I won't mention names, but some are worth 80, 100 times what we are, and they're still not just with either a very small percentage of the market in the colorectal space or a small percentage in the DNA space. And we absolutely passionately believe the nucleosome itself is an incredibly important part of what makes us tick, and we absolutely dominate the space in circulation. So if that is true, the epigenetics are as important as the genetics. The next few years are going to be spectacular for us as we roll out the products and all these different things going on, which we will continue to do on a very, very low cost, very focused on the platform.
I do like the opportunistic approach that this company has in terms of the low cost base, in terms of the way they do the trials, the way they do the research, the way they set up, and also, you know, the approach they have now in terms of trying to chase some revenue on the veterinary side. So, yes, I'm there for the long haul.
The first time we hear about the veterinary market, first time we hear about the Nu.Q Capture, which I think is really exciting. So personally, I really enjoy, you know, here the new opportunities of Volition, and the fact that it's a platform that can grow outside just one cancer diagnostic gets me as an investor very excited.
It's a totally differentiated technology from what everyone else out there in the blood-based biopsy space is doing. As you heard today, a lot of the focus has been on looking for specific mutations in DNA and trying to find these very difficult to spot features. Now, Volition's approach, focusing on the circulating nucleosomes, is very different, and so that's something that attracts us to the technology. We think it could be used as a standalone technology or in conjunction with some of the existing technologies that are being developed by companies like Grail, like Garden. They have an excellent platform technology that is still being fleshed out, and I'm looking forward to seeing what happens with it.
As I said earlier, all kinds of people have been attending today's presentation by Volition RX, including key opinion leaders and experts.
Good afternoon, everybody. My name is Jake Saad, is Axel Imhof. I'm a professor at the University of Munich, and I'm the last addition to the scientific advisory board of Volition. And what
I'm actually very excited about joining Volition, and especially about this Nu.Q Capture assay that they're developing, because this is something which I think has the great potential to actually look for new biomarkers and look how we can even improve cancer diagnostics by using epigenetic features and then describing it much better than what is now possible with the Nu.Q technology, which already is quite good.
The current companion animal space receives about twice as many cancer cases than in the human market. And currently the process for actually determining cancer is a very complicated and timely process where it's weeks on end, and it's a sandwich approach of many tests being done. The ability to do a small sample and get results within a few days using Nu.Q is going to bridge a very large gap that currently no assay or test is currently filling.
Quite amazing that the curve we've been on. The first few years were in my lounge room in a very small lab in Belgium, and then a slightly bigger lab of a few hundred square foot. And then now the team we have is over 45 people, and we have a 20,000 square foot facility in Belgium, really making sure we do the groundwork to make the platform really viable and that can be used in many, many cancers, and even on dogs, and even for Nu.Q Capture.
It's been a great day, not just meeting shareholders and seeing more opportunity in what Volition is actually doing and how that's really going to change the marketplace, not just on the dog side, but also on the human side.
I'm a very academic researcher, so for the last, I don't know, 25 years or so, I've been working in chromatin. And I think now it starts to get into an area where all of a sudden, you know, people profit from it, and you know, you can envision a diagnostic tool that is based on a lot of the research that I've been doing the last 25 years, which was very basic. And you know, now that seeing that translated into the clinics makes me very excited.
So I'm ending this special report on Volition RX here in New York on the trading floor itself. And as we've heard, there's a lot going on with the company right now, but also some important milestones coming up in the future. This is Nick Wood at the New York Stock Exchange saying bye for now.